Hello dear learners, welcome to my channel Embrace Biota. This is the part 2 of our cell biology. पार्ट 1 में हमने सेल बायोलॉजी के बेसिक्स, सेल वॉल, सेल मेम्ब्रेन और दो ऑर्गेनेल्स को डिस्कस करा है. That is mitochondria and plastids. In today's video, we will cover the rest of the cell organelles. We are starting with the nucleus. The nucleus, also known as karyon, is discovered by Robert Brown in the orchid root. Study of nucleus is known as karyology. This karyon word is used for the nucleus and the terminology related to the nucleus. The nucleus is the control center of the cell, also known as the heart of the cell, and is the director of the cell. Hammerling performed the experiment on the nucleus by using the species of the green algae Acetabularia and conclude that the nucleus contains the chromosome and genes, so is the controlling center of the cell. The nucleus consists of the nuclear membrane, nuclear pore, nuclear lamina and nucleoplasm. The nuclear membrane is divided into the outer nuclear membrane and inner nuclear membrane. The outer nuclear membrane contains the ribosomes attached to it and extend to forms the endoplasmic reticulum. The space between two nuclear membrane is known as perinuclear space. The nuclear membrane is a continuous structure, but its continuity is broken by a minute pore known as nuclear pore. Nuclear pore is discovered by Collin and Tomlin. The nuclear pore consists of the 8 cylindrical protein known as annulus, which forms the nuclear pore complex. This nuclear pore complex is seen only in the animal cells. Between the nuclear envelope, there is a filamentous network of protein known as nuclear lamina. It is made up of intermediate filament lamin A, V, and C. Function performed by this lamina is to attach the chromosome to the nuclear envelope, and also it helps in the growth of the envelope. Then we have the nucleoplasm known as the nuclear sap. This nucleoplasm consists of the nucleolus and chromatin. Nuclear pore के बारे में और जाने तो it helps in the bidirectional movement of RNA and protein. It is the movement of RNA and protein from the nucleus to the outside and from outside to the nucleus. It is made up of the 8 annulus ring-like structure which forms the nuclear pore complex. This complex helps in the regulation of diameter of nuclear pore. The nucleus is absent in the mature RBCs, sieve tube cells, and prokaryotes. On the basis of the number, it can be uninucleate, binucleate, or multinucleate. Most of the plants and animals are uninucleate. Binucleate nucleus is seen in Paramecium caudatum. The multinucleated condition is known as syncytium or cynocytic. In animals, it is found in the striated muscles and opalina. In plants, it is seen in the Baucaria and the endosperm of coconut. The nucleus is also variable in shapes. The variable shapes are present in the WBCs. In neutrophil, there is multilobed nucleus. Lymphocyte have spherical nucleus. In monocytes, it is kidney-shaped. In the eosinophil, there is horseshoe-shaped nucleus. And in basophil, it is bilobed and appear S-shaped. अगर हम न्यूक्लियोप्लाज्म को डिटेल में देखें, it consists of the nucleolus and chromatin. The chromatin is composed of the DNA and the histone protein. And the nucleolus was discovered by Fontana. Nucleolus is a non-membranous structure and is always at the periphery of nucleus. It helps in the synthesis of ribosomal RNA. This nucleolus formed at the NOR region of the satellite chromosome, and the chromatin fiber is stained using fusion stain. There is usually one nucleolus per cell, but the number of nucleolus depends upon the cellular need. If the cell is metabolically more active and participate in the protein synthesis, then the number of nucleolus may be more in that cell. And if it is less involved in the metabolic activities, then the number of nucleolus are less. After nucleus, हम बात करेंगे एंडोमेम्ब्रेन सिस्टम की. It is also known as GERL system. It includes Golgi apparatus, endoplasmic reticulum, lysosome, and vacuole. The function of all these organelles are occur in coordinated manner. First organelle of this system is endoplasmic reticulum. The endoplasmic reticulum was first observed by Garnier and called it as ergastoplasm. But the main credit for the discovery of ER goes to the Porter. It was Porter and Claude who first observed the ultrastructure of endoplasmic reticulum. Endoplasmic reticulum is the extension of nuclear membrane. It is a network-like structure and is scattered in whole of the cytoplasm and divides cytoplasm into two components. The components which comes inside the lumen of endoplasmic reticulum is known as intraluminal component, and the part outside the lumen of the endoplasmic reticulum is known as extraluminal component. It is also called as cytoplasmic compartment. अगर हम स्ट्रक्चर देखें एंडोप्लाज्मिक रेटिक्यूलम का, इट इज मेड अप ऑफ 3 पार्ट्स. फर्स्ट वन इज द एंडोप्लाज्मिक सिस्टर्ने, second is tubules, and the third is vesicles. The cisternae are unbranched structure, and the rough endoplasmic reticulum is made up of cisternae. It also contains the ribosome, whereas the tubules are branched and it do not contain the ribosome. So the smooth endoplasmic reticulum is made up of tubules. The vesicles are the rounded structure and filled with the chemicals and proteins. These vesicles fuse with the cis phase of Golgi complex. On the surface of rough endoplasmic reticulum, the ATS ribosome is present, whose subunits are 40 and 60S. And the most important thing is that these ribosomes attach to the surface of RER with the help of ribofourin protein. This protein attaches the 60S ribosomal surface with the endoplasmic reticulum. We have two main types of endoplasmic reticulum. First is rough endoplasmic reticulum, which contains the ribosome attached on it. Due to the presence of ribosome, this reticulum is known as granular endoplasmic reticulum. RER is about 2/3 part of the endoplasmic reticulum. It is unbranched and made up of cisternae, help in the protein synthesis. Second is smooth endoplasmic reticulum, which do not contain ribosome attached on them. The smooth endoplasmic reticulum due to this is known as agranular endoplasmic reticulum. It covers only 1/3 part of the endoplasmic reticulum. The SER is branched and made up of tubules. It helps in the lipid synthesis and synthesis of the steroidal hormones. RER is proteinaceous, so is more stable than SER. Rough endoplasmic reticulum in the nerve cells modified to form the Nissl granules. The Nissl granule is the modification of rough endoplasmic reticulum. RER present abundantly in pancreatic cells, liver cells, and nerve cells, whereas the SER is dynamic structure as it is composed of the lipids. And in the skeletal muscles, the SER modified to form the sarcoplasmic reticulum. So the endoplasmic reticulum of the muscle is known as sarcoplasmic reticulum. It is the storehouse of the calcium ion. SER is abundant in muscles and adipose tissue. In addition to these two types, there are some special forms of endoplasmic reticulum. First one is the desmotubules. This is the part of endoplasmic reticulum which passes through the plasmodesmata. And the plasmodesmata is present only in the plant cells, so these desmotubules are also occurs in only plant cells. It establishes a transport system between the two cells. Another modification is microsome. When we centrifuge the fragmentation of ER, The vesicles-like structure is formed known as the microsomes. बात करें functions of endoplasmic reticulum की, it helps in the protein synthesis with the help of RER. And the SER component helps in the synthesis of cholesterol, steroids and steroidal-like hormones. Also, the fat-soluble vitamins are synthesized by SER. Most important function is that the vesicles of ER gets flat and forms the Golgi complex. SER helps in the synthesis and storage of glycogen. Another notable point is that the myeloid body in the retina is the modification of SER. In SER, the cytochrome P450 is present. It helps in the detoxification of inner tissues. SER also helps in the formation of microbodies like peroxisome, glyoxysomes and spherosomes. After endoplasmic reticulum, we have Golgi complex. Golgi complex, also known as Golgi apparatus or Golgi bodies. It was discovered by Camillo Golgi in Purkinje nerve cells of the cerebellum of cat and all by staining with the silver nitrate. And he coined term Golgi apparatus for them. In plants, this apparatus is found in the form of the single cisternae, so is known as dictyosome. This complex is also called as the Dalton's complex, lipochondria and the Baker's body. The maximum Golgi complex is seen in the rhizoidal cells of the Cara algae. It is also known as traffic police of cell. It arises from the vesicles of smooth endoplasmic reticulum. Golgi complex is also known as idiosomes. There are two faces of the Golgi body. First is cis face and second is trans face. The cis face is also known as the convex face, proximal face or forming face. And the trans face is known as concave face, distal face and maturing face. The cis face is close to the nuclear membrane. The vesicles of endoplasmic reticulum fuse with the cis face. The trans face is towards the plasma membrane. Lysosomes arise from the trans face of Golgi body. The Golgi complex consists of the Golgian cisternae, Golgi vesicles and the Golgi vacuole. The cisternae is the functional unit of Golgi body. It is formed by the SER. Each cisternae is separated by the intercisternal space. At the corners of the cisternae, the vesicles are present. The vesicles pinches off from the cisternae and forms the Golgian vacuole. This vacuole forms the primary lysosome after detachment from the trans phase. Golgi body is the only organelle which shows the polarity, that is having the cis and trans phase. And the important thing is that the vesicles of the Golgi pinches off and forms the vacuole. The function of the Golgi bodies are: it helps in the recycling of plasma membrane, also helps in the formation of cell wall. It forms the primary lysosome. It is the site of processing and modification of proteins. It helps in the packaging of materials. Also forms the acrosome of the sperm head. By the process of glycosylation, it forms the glycoprotein. And by glycosidation, it forms the glycolipid. It stores the vitamin C and help in the sulfur metabolism. The marker enzyme of Golgi bodies are glycosyltransferase and manosidase. After Golgi complex, we have lysosome. Lysosome is known as suicidal bag, atom bomb, scavenger and disposal system of cell. The discovery of lysosome is done by D. Dube. It is the single membranous structure which is present in all animal cells except the mature RBCs. The primary lysosome are formed by the packaging process of cis phase of Golgi body. Due to the presence of high concentration of H positive, the lysosomal pH is acidic. It contains the hydrolytic digestive enzyme for the digestion of carbohydrates, proteins, lipids, and nucleic acid. Lysosome play a role in the intracellular digestion of particles. It is the polymorphic cell organelle, so exist in the many forms. Various forms of the lysosomes are: first is the primary lysosome, also known as true lysosome or proteolysosome. This is the lysosome formed by the Golgi body. It is filled with the inactive enzymes. The secondary lysosome is also known as digestive lysosome. When the primary lysosome fused with the target cell, it forms the secondary lysosome and it is filled with the active enzymes. And the digestion of the target organism gives rise to the tertiary lysosome. It is also known as residual body or the teleolysosome. Another type is the autophagic lysosome, which performs the digestion of the surplus or the waste cell organelles. Talking about the function of lysosome, lysosome performs the heterophagy, that is digestion of the foreign particles like bacteria and viruses. Lysosome also performs the autophagy. It means the digestion of on-cell organelles like mitochondria. It also performs autolysis. It means it releases its own content and help in the bursting of the cell during the damage. Due to this reason, it is known as the suicidal bag of the cell. The marker enzyme of lysosome is acid phosphatase. Staining of lysosome is done using Gomori stain. And the substance which stabilize the membrane of lysosome are cortisone, also known as hydroxycortisone. And the substance which weakens the membrane of lysosome are called as the levelizers. It includes digitonin and bile salts. Our next organelle is vacuole. Talking about vacuole is a single membranous organelle. In plants, it was discovered first by Anton von Leeuwenhoek. Plants के केस में vacuole is large and occupies the central surface. And in animals, the vacuole are emerges as a part of endoplasmic reticulum and Golgi complex. So many vacuoles are present in animal cells and are smaller. The large central vacuole in the plants covers almost 90% of the cell volume. Due to this, the nucleus and cytoplasm comes in the peripheral side. This condition is known as primordial utricle. Most important thing is that the plasma membrane of vacuole is known as tonoplast and the cytoplasm is known as cell sap. This cell sap is the non-living structure and it contains the waste and excretory material of the plant. The cell sap is also rich in the anthocyanin pigment. Anthocyanin pigment is purple color pigment and is water soluble. अगर कुछ स्पेशल टाइप्स के वैक्यूल की बात करें, वी हैव कॉन्ट्रैक्टाइल वैक्यूल. These are seen in protozoa like amoeba and paramecium and meant for osmoregulation. Food vacuole is seen in amoeba. And most important is the gas vacuole. It is feature of blue-green algae and purple sulfur bacteria. This vacuole plays the role in buoyancy. The last organelle of today's discussion is ribosome. The ribosome was first discovered by Claude in the microsome. Microsome is the fragment of endoplasmic reticulum. Ribosome is the non-membranous cell organelle. It is the smallest cell organelle known. It is mainly found on the endoplasmic reticulum and inside the nuclear membrane. So we called it as the organelle within the organelle. Ribosome is known as the protein factory of the cell. These are discovered by pallid, so also known as the pallid granule. Ribosome in plants was observed by the Brown and Robinson. Ribosome is a ribonucleoprotein. It means it is composed of the nucleic acid as well as protein, so called as nucleoprotein. Nucleic acid here is the rRNA. The protein is known as R proteins. In prokaryotes, the ratio of rRNA and R protein is 60:40, whereas in eukaryotes, this ratio is 50:50. RER is present in both prokaryotes as well as eukaryotes. In eukaryotes, it occurs in the free form in cytosol, mitochondria, and plastid, whereas also occurs in the bounded form and is winded with the ER or the outer nuclear membrane. Ribosome in eukaryotes and prokaryotes is made up of different subunits. In eukaryotes, the ribosome is 80S type and in prokaryotes it is 70S type. The 80S ribosome is made up of 4 different types of subunit. Smaller subunit is 40S and the larger is 60S. The 40S subunit contains the 18S rRNA, whereas in 60S, 5S rRNA, 5.8S rRNA and 28S rRNA is present. In prokaryotes, the 70S is made up of 3 different types of subunit. The smaller subunit 30S has the 16S rRNA. In larger subunit 50S, the 5S rRNA and 23S rRNA is present. In plant mitochondria, the ribosome is 70S type and in animals it is 50S type. In the prokaryotes, the several ribosomes are held on the string of one mRNA known as the polyribosome, and in eukaryotes is known as monosome. There is only one ribosome on the string of mRNA. Both the subunits of ribosome are joined to each other with the help of magnesium ion. The high concentration of the magnesium facilitates the association of subunits, and the low magnesium concentration dissociates the two subunits. The S written with subunits indicates the sedimentary coefficient or Swedberg unit. This coefficient is the indirect measure of the density and size. The value of Swedberg unit is 10 raised power minus 13 second. The 3 sites are present in the ribosome known as EPA site from the 5' to 3'. E stands for the exit site, P is the peptidal site and A is the amino acyl site. And the most important thing is that the smaller subunits bind with the mRNA during the protein synthesis, whereas the larger subunits bind with the tRNA during protein synthesis. The ribosome staining is done using toluidine blue stain. This is all about today's video. In next lecture, we will discuss about the cytoskeleton and microbodies. If you like the video, subscribe the channel. My name is Anna Sharma and thanks for watching.